What actually happens when your brain tells your muscles to move? The signal has to jump from a nerve to a muscle, and the place it does that is a specialized synapse known as the neuromuscular junction. So that's a junction between your nerve and your skeletal muscle. Yo, what's up YouTube? It's Holistic Jordan. I'm a first year medical student, and in this video I'm going to be talking about the anatomy and the function of the neuromuscular junction. Okay, be sure to tap in, and I'm going to get straight into today's video. So yeah, the neuromuscular junction is a specialized synapse between a neuron and a muscle, and it has three main parts. Okay, the first part is your presynaptic nerve terminal, and that is the end of the motor neuron. So here we have your axon terminal, which is going to split into your synaptic end bulb. Okay, so that's that little bulb shape. That is the end of the motor neuron, and that's the first part of your neuromuscular junction. Okay, the second part of the neuromuscular junction is your synaptic cleft, and all that is literally just a space between your motor neuron and the start of your skeletal muscle. Okay, the third part of your neuromuscular junction is your motor end plate. So that's kind of like a specialized region of your sarcolemma, and your sarcolemma is literally just the membrane of your muscle cell. So let's focus on your synaptic end bulb. Okay, so you have these circular structures called vesicles. Okay, and vesicles are containing an important neurotransmitter, and that is ACH, also known as acetylcholine. But let's take a step back. So we know that for information has to travel from your motor neuron to your muscle to communicate to the muscle to make it contract. Okay? So coming down your axon terminal, you're going to have a wave of depolarisation. Okay? So you're going to have that influx of sodium into the motor neuron. And what is that going to do? Okay? So over here, these are called your voltage-gated calcium channels. So just abbreviate it, voltage-gated calcium channels. Okay? And what's going to happen is, so when we even think about calcium, this is an extracellular ion. Okay? So the concentration of calcium is higher outside of the cell than inside of the cell. So when we have that wave of depolarization moving down the axon terminal, you are going to activate your voltage-gated calcium channels, and because the concentration of calcium is high outside of the cell, what do you think is going to happen? That's right, calcium is going to come inside the cell now. So it's going to move through the voltage-gated calcium channels, and it's going to enter into the cell. Okay, but what does calcium have to do with muscle contraction? Now that we have calcium inside of the cell, what does the calcium do? So calcium is going to be in direct communication with your vesicles containing acetylcholine. So what calcium actually does, it causes the vesicles to move further and further down your presynaptic nerve terminal. Okay, so if I just get a pen, where is my other pen? Okay, so let's say this one, oh, that pen's not working. Okay, found a pen. Okay, so let's just draw it out. So these are your acetylcholine in blue, and your vesicle black. Let's write vesicle. Okay, so the calcium is going to cause your vesicle containing acetylcholine to move down to the postsynaptic nerve terminal to dock, and it's going to fuse and release its contents via exocytosis. And exocytosis is bulk transport outside of the cell. So the vesicle is packed with acetylcholine, and it's going to diffuse. It's going to release content via exocytosis and then diffuse across the synaptic cleft for its destination. Okay, so in the synaptic end bulb, we're going to get a rush of calcium coming into the cell via voltage-gated calcium channels. Once it reaches the level of depolarization to activate the voltage-gated calcium channels, calcium comes inside of the cell. It's going to cause your vesicles containing acetylcholine to move to your presynaptic nerve terminal and to release its contents via exocytosis, where it releases the acetylcholine into the synaptic cleft. Okay, so that's where we are at this point. Okay, so now let's talk a bit about the synaptic cleft. We already know that this is just a space between a motor neuron and a muscle cell, okay? So what's going to happen is that once the acetylcholine is released into the synaptic cleft, it's going to diffuse closer towards your motor end plate. So now we're coming into your postsynaptic membrane. This is where acetylcholine is going to meet your muscle, okay? And a couple of the specializations of your sarcolemma, so the membrane of your muscle fiber, is that it has a region of high folds. Okay, it has a lot of folds to increase the surface area. Okay, and not only that, it has your nicotinic acetylcholine receptors. Okay, so my pen is slightly giving way. Yeah, let me get that pen. Okay. Yeah, the red pen does it. Okay, so you have your nicotinic acetylcholine receptors. Just try to abbreviate that. And with your nicotinic acetylcholine receptors, they are complementary to acetylcholine. So when acetylcholine binds, when two acetylcholine molecules bind to your acetylcholine nicotinic receptors, it causes a conformational change within the nicotinic receptors, okay? And a small pore opens up. And that small pore allows sodium. So let's say we have sodium here. Sodium has a high extracellular concentration. Okay, so there's a lot of sodium outside of the cell. Okay, outside of the muscle cell. Okay, and that small conformational change is going to allow sodium to then influx, so to come inside of the cell and to depolarize the motor end plate of the cell. And that's how you eventually get muscle contraction. But hold up, I said that two acetylcholine molecules bind to the nicotinic receptor to cause a conformational change. But what happens when we don't want the muscle to be contracted? Because if acetylcholine is constantly bound to the nicotinic receptor, your muscle is going to stay contracted, and you don't want your muscle to always be contracted. So there's a specialization within the synaptic cleft which causes the breakdown of acetylcholine, and that's your excitatory neurotransmitter, which is going to stimulate that muscle contraction. Okay, and that enzyme, which is going to break down acetylcholine, is your acetylcholine esterase. Okay, I'll write that out fully as well. Acetylcholine esterase. And what that's going to do, once acetylcholine is bound, acetylcholine detaches from your nicotinic receptor, and your acetylcholine esterase breaks it down into acetate and choline to basically prevent further muscle contraction. So across the cell, the motor end plate is a specialized region of your sarcolemma. Once you have sodium coming inside of the cell now, okay, the cell becomes more depolarized, and you're going to reach activation threshold where you're going to activate voltage-gated sodium channels. So you're going to have even greater depolarization coming inside of the cell. Okay, so that depolarization is going to cause an action potential to propagate throughout your muscle, and you're eventually going to get muscle contraction. So just recapping this video, the neuromuscular junction is a specialized synapse between your neuron, your motor neuron, because it's containing those information to move your muscle, and your muscle. Okay, neuron meeting muscle. The muscle meets the neuron. The neuron meets the muscle, but the muscle is communicating with your. Okay. Hopefully that makes sense. Also, your synaptic end bulb is going to release acetylcholine from the stimulus of calcium influx coming inside the cell. Acetylcholine is going to move towards the nerve terminal dock, and it's going to release its contents via exocytosis. So now acetylcholine is in the synaptic cleft, that space between the motor neuron and the muscle. In your sarcolemma, these specializations have a lot of folds and a high concentration of the nicotinic acetylcholine receptors, which allows acetylcholine to bind, cause a conformational change, get sodium influx. You're going to reach threshold, and you're going to get your activation of your voltage-gated sodium channels open, and to get more influx of sodium, and it's going to depolarize the cell. The main aim to contract the muscle. Okay, so that's the video. Hopefully this helps you visualize what's going on at the neuromuscular junction. And the main takeaway, the neuromuscular junction has three parts: presynaptic nerve terminal, the synaptic cleft, and the postsynaptic membrane, also known as the motor end plate. which is going to depolarize, eventually leading to muscle contraction. Okay, if the breakdown helped, be sure to like, subscribe to the channel, because I will be coming with a lot of medical teaching videos and insights into like the things I learn about to keep me afloat, making sure that I'm revising my content as I continue my journey throughout medical school. Doing whiteboard teachers like this, it helps me understand it. I'll do my best to break it down in a really simple, easy way to help other students interested in medicine or anyone just really interested in science. It's Holistic Jordan. In one of my next teachers, I'll talk about what happens after acetylcholine binds. Talk about the whole actin, myosin, sliding filament theory, all of that. Okay? So, it's Holistic Jordan. I'll see you in the next video. Peace.